Hi everyone, and welcome to our first microbiology lab. Now, I know everyone would much rather be in the lab itself, having the hands-on experience, but while we are in the online setting, I will try to make these as, painless and as informative and valuable as possible. Now, as always, if throughout the lesson you have any questions or concerns, feel free to contact me in the Remind app. Now, the first thing we're going to start off with today is the idea of lab safety, which is very important for a microbiology lab because this course is a BSL2 lab. Okay, so I want you to circle, star, highlight the term BSL2. Okay, we're going to go through in the coming slides what that actually means. But what I did here on this slide is I listed out a whole bunch of questions to guide you, basically questions that by the end of our lab safety discussion in the first part of this lesson, you should be able to answer all of these questions. If there's any that you are not able to answer at the end of this lesson, please again contact me in the Remind app and I will help you along. Now, one of the terms you have to be familiar with in this class is PPE. Now, thanks to the COVID crisis, a lot of students now are aware of what that means, and a lot of the public know what that means now. So it stands for Personal Protective Equipment. Now, during the COVID crisis, most people think of that as wearing a mask and wearing gloves. In our laboratory, the two main examples of PPE that you always need to have, whether there's a pandemic or not, are your lab coat and your gloves. Okay, very important, and it's very important that both fit you well also. Okay, whenever you grab gloves in a lab, make sure that you wear the correct size because incorrect ones can end up being very dangerous to you. If they're too small, they could end up ripping and you get exposed to something you don't want to be exposed to. And if they're too big, they tend to get caught in various things. So for instance, the little microcentrifuge tubes that you use in experiments, sometimes you end up closing an oversized glove in that kind of a piece of equipment, and it ends up causing problems for your experiment. Now, there are various rules that I want you to keep in mind when it comes to your lab coat and gloves. Anytime you are in a lab, when you are wearing these things, there are certain places they are to be worn, certain places they're not to be worn. So, where should they be worn? In the lab, okay? Always wear them in the lab. Where should they never be worn? Anywhere that is not the lab, okay? That means you never step into the hallway, into any other room while wearing a lab coat or gloves, okay? They are to remain in the laboratory, and ideally, you would even store them in the lab that you're working. Okay, so for instance, at our school, we give you lockers to store them, and we then overnight put them in the laboratory in sealed containers. Okay, you want to make sure that they are kept safe and away from your other belongings and whatnot. Now, when you are wearing PPE, particularly gloves, you have to ask yourself, well, what do I have to be careful in terms of what I do not touch while wearing gloves? You never want to touch doorknobs, any kind of handles that other people use when they're not wearing gloves. And you also never want to touch any of your personal belongings. Now, unfortunately, as a culture, we have become glued to our cell phones. And I know it's a lot to ask for you to not touch those cell phones for the, 1 to 3 hours that you're in a laboratory setting. But you have to be very careful when it comes to those rules, because you want to keep all contamination off of you and your belongings. And you never want to track anything home to your loved ones or to anyone that you encounter outside of the laboratory. So please pay close attention to making sure that all contamination stays in the laboratory and does not go elsewhere. Once you are done with any experiment, you always want to make sure to dispose of gloves in the properly labeled biohazard garbage pails. Now, we also have a lot of clothing safety restrictions, because even though you are wearing a lab coat and you're wearing gloves, if you're wearing the wrong kind of clothing, you end up putting yourself at risk as well. So the first big rule is never wear open-toed shoes, and that means even if you think, oh, I have socks on, that's fine. No, you never want to wear open-toed shoes. The example I give is back when I was a PhD student. I had been working in labs for a very long time at that point. I was very experienced. It was summertime, and I was doing an experiment where every few hours I had to return to the lab to check on my cells, the cultures that I was working on. So one of the last readings was at 11 p.m. at night, and I didn't realize that it was already close to that time. I was out and about because it was summertime, and I had sandals on. But I figured, well, it's 11 o'clock at night, no one else, summertime, no one else going to be in the lab. It's okay if I just run in quick. I knew what I was doing. All I had to do was pipette a little sample into a special machine, read it, I'm done. So I went in and I did the experiment anyway while wearing sandals. Now, I was ultra careful with every step that I was taking, with every piece of equipment I was handling. What I did not know is that even though I was being really careful, other people who had worked in the lab earlier that day were not so careful. Turns out one of the other students had broken the beaker, the glass container that we use as a waste bin, and rather than telling anyone or properly disposing of it, they just turned it in a way that you couldn't see that there was a big hole on the bottom. That meant that when I went to move it to do my part of the experiment, all of the waste, all of the gross, used up cells and cultures and nasty media fell down right onto my foot. Okay, and because I was wearing sandals, I then had to do crazy protocols of proper cleaning, and I lucked out that I was not seriously injured. But I could have gotten very sick from that, okay, because you never know what other people are going to be doing in a lab. You can be the most careful person in the world, but you don't know what those around you will be doing. So always make sure to be careful about wearing properly closed shoes. Never having exposed skin, whether it's your legs or your arms, because again, your lab coat's not going to cover everything. So make sure to wear proper clothing. And if you have long enough hair, make sure that it's tied back, because hair can easily end up in any of your experiments. You don't want, for instance, if you're working with bacteria, for your hair to flop into your bacterial plate or culture, and then you track that home with you or everywhere you go. And you work with things like Bunsen burners. And so you do not want to set your hair on fire, okay? So always be very careful. And I know you're thinking like, ugh, why is she going through this? You know, we know not to do that, or that never happens. Well, newsflash, these things that we go through, we have to tell you, because it means somewhere along the line, at some classroom, at some school, bad things have happened when students were not careful. And that's why we end up with lists like this to try and protect you. Now, in addition to making sure that you have proper PPE and that you're being careful of contamination, you have to keep in mind that two of the biggest causes of lab contaminations are food or drinks, as well as cell phones, which I've already mentioned in the earlier slides. So whenever you are dealing with any lab, whether it's microlab or any other labs, always make sure that you do not have any food or drinks anywhere near your experiments. If you have a bag with you, backpack, locker, depending on what lab you're in, always make sure that your food and drinks are away from your lab, stored somewhere else. In my class, in terms of the labs, you are always more than welcome to step out into the hallway to have your snack or to have a drink of water, but you cannot have any of that in the laboratory itself. Also, make sure to never have your cell phones anywhere in the lab. Okay, they have to be kept away. Again, I know that's brutal to ask people nowadays that we are, addicted to our cell phones, but trust me, you do not want to have any bacteria that we are working with end up on that cell phone, where your hands are touching it all day, where you're putting it near your face, and where you're bringing it home to where your loved ones are. Now, please keep in mind that if we do make it to the in-person weeks, you are handling actual bacteria, okay? The tubes we give you may look like, lemonade or some beer, the coloring, but it's not. It's actually bacteria in there, and they are alive. And if you are not careful, they can cause you harm. Now, again, because we are a BSL-2 lab, you will be working with organisms that have the potential to make you ill, but for which there are, readily available treatments, okay? But you still do not want to get any of these bacteria on you, on the lab benches, anywhere. So please be careful about not shaking any of the tubes that you have that can create aerosols, fine particles throughout the air that you would breathe in, that can also create spills that become problematic. Okay, always make sure that you are very aware of your tubes, of where the bacteria are, about not letting them spill or touch any of your belongings or anything you're working with. And always make sure to keep any bacteria tubes on a rack, especially when you are moving them from one location to another. If you do spill, then you want to immediately grab disinfectant, spray it on the spill, and let it sit for at least 15 to 20 seconds. Okay, you want it to sit on there, let it do its work of killing the microbes. Then you want to wipe it up nice and clean. You then want to make sure that when you dispose of those paper towels, you're putting them in the biohazard container. And what else do you have to get rid of? Your gloves that you are wearing, because they could have gotten contaminated now as well. So make sure to throw out those gloves in the biohazard pail and then put on a fresh pair. Now, this slide is very important when you're working in any microbiology lab. Always make sure to spray down your benches before and after you do anything. Okay, so you spray them down with whatever disinfectant the classroom has available. Sometimes it's Lysol, Clorox, or just a bottle that generically says disinfectant. But always make sure you spray down those benches at the beginning and end of every class. We do it at the beginning also because when you walk into these laboratories, you don't know how careful the people who were in there before you were. So it's best to be safe and careful yourself, rather than just relying or assuming, oh yeah, I'm sure the other people, they cleaned it up, I should be fine. Nope, always spray it down yourself before and after. Also, always make sure to wash your hands at the end of every lab, even though you were wearing gloves, okay? Proper hand washing in settings like this is so important because hand washing at all is the most effective way to break the chain of infection. And you've seen that now that you've all lived through a pandemic and you've kind of gotten firsthand experience of why hand washing is so valuable. So now the last thing I want to mention when it comes to lab safety are two terms I want you to be familiar with. They are aerosols and BSC. Now we already mentioned aerosols. That's a fancy scientific way of saying that a solid or liquid has basically become these fine particles that you can inhale. So whenever you're handling bacteria or any kind of sample with living organisms, you want to make sure not to shake or vortex them, because then you can create aerosols. You breathe in these fine particles and now you can become infected. The other term I want you to be familiar with is a BSC, which stands for Biosafety Cabinet, which you see in this picture right here. Now, Biosafety Cabinet, that's basically a sterile workplace that helps to protect the worker, which you see here, as well as the rest of the lab environment from whatever you're working on. It also helps protect the experiment itself from what contamination may be in the environment as well. Basically, when you're going to work with one of these, you make sure to turn it on right before you start your experiment to disinfect everything in there. You then can leave it on during your experiment and make sure to have it on for a little bit after to make sure all the surfaces and all of the items get sterilized or disinfected as well. And we're going to go over that kind of terminology more in the lecture. Now, I want to point out that when you're doing microbiology experiments, a lot of times people will work with metal inoculation loops to grab the bacteria and to put it on plates or to perform their experiment. And the way they sterilize these loops before and after they use them is usually with a Bunsen burner, so an open flame. Now, I want you to put in big writing on this slide that you can never use a Bunsen burner to sterilize your equipment when working with a BSC. Okay? There are 3 reasons you never want to use a Bunsen burner or an open flame with a BSC. One of them is, of course, it's a fire hazard. Okay? It can be very dangerous, especially if the flame gets out of hand or gets knocked over in there. The second reason is that it also damages the filters that keep that BSC working properly. And third, it disrupts the airflow. Again, part of what makes that cabinet work properly is its proper ventilation and airflow. And having a flame in there messing with the oxygen levels can greatly disrupt that. And like I just said, can also damage the filtration of that unit. Okay, so you never ever want to use a Bunsen burner. Instead, what we use in there are disposable plastic loops or spreaders, as well as what we call Bacti incinerators. And an incinerator is basically an electric way of getting that loop that you're using really, really hot and sterile. Okay, so please keep these terms in mind once we hopefully make it into a lab setting. Now that's enough about the lab safety. We'll revisit some of that information when it comes closer to the possibility of having some in-person laboratory classes. For now, what I want to do is move on to the microscopy review, and you've probably encountered most of this in your other classes, so I'll kind of run through it rather quickly. But again, if I go through anything too quickly or you need some practice problems or any extra information, please contact me in Remind. Never hesitate to contact me in the Remind app. So in a microbiology lab, the kind of microscope that you would encounter is a compound or a light microscope. And basically the function of these microscopes is that they use lenses to bend light rays in order to bring an image to your eyes. So whatever image the slide that you're looking at has. Now, the two lenses that will usually be involved are the ocular lenses where you'll put your eyes, and then the objective lenses down at the bottom of the nose piece, which we'll review in a minute. Now, basically, when the light is going through those lenses in order to get to your eyes, you'll notice that the light rays end up crossing. So between where the specimen is, that microscope slide, and your eyes, the light rays end up crossing once, okay? When those light rays cross, what it ends up doing is it flips the image that you're seeing. So the image ends up upside down and reversed. Now, if you ever look through a microscope and an image is not upside down and reversed, that means that the light rays have crossed a second time to correct for that flipping. And what it specifically means is that there is a third lens in that microscope. Okay, so if I ever ask you, what does it tell you if the image is not upside down and reverse, instead it's normal, it tells you that there's a third lens in that microscope. It's called the projector lens. So you'll find it between the ocular and the objective. So that now when the light rays cross the first time, it flips the image, and then they cross a second time to flip it back to normal. So if we were to draw out this concept, I'm just going to switch over from the laser to the pen. Okay, so one of the slides we usually have students look at is a slide where you have the microscope slide and it has a letter E in the center. Okay, I'm sorry, I'm drawing over my keyboard, so it's making it a little shaky and weird looking. So that's how it looks on the slide. When you look at it in your microscope, what you end up seeing is the letter E, but it's upside down and reversed, okay? Because that is what we talked about over here with the crossing of the light rays and the image flipping over. So when you look at an image in a microscope and it's upside down and reverse, that tells you there are two lenses. Whereas if you look at that slide and you see the letter E perfectly normal, what does that tell you? It tells you there are three lenses, specifically that there is a projector lens. Okay? And that question tends to come up, so make sure you circle star highlight that fact. And that's why in a lot of classes when you review microscopy, you're asked to look at the letter E slide because it shows you that the microscope you're using only has two lenses. The light rays cross over, it flips the image, as you can see right there. Okay? Whereas if it looks normal, you have three lenses, including a projector lens. If there's any difficulty understanding that, again, please send me a message in Remind app and I will further clarify. Now, whenever we talk about the microscope, it's important for you to review the parts, and I want you to be able to quickly identify the purpose of any part. So I can either ask you what is the function of the stage, or I can describe a function and you would have to tell me, hey, she's talking about the stage, for instance. So the first part we review is the stage of the microscope, which you see right over here zoomed in, right over here in the further out picture. And the purpose of this stage is to serve as a place for you to put the microscope slide. So you would just place the slide right here. Now the stage also has what's called an aperture. This hole right here, that is the aperture. Now notice the aperture is not something that is changing shape or size or anything. It is just simply a cut out opening in the stage because its purpose is to let light through. Okay, so the purpose of the aperture is to let light through to reach your slide. Now, the aperture cannot control or modify itself. Instead, if you want to control or modify the amount of light that's reaching your specimen, you then have to adjust the condenser, okay, which has a diaphragm which can open or close. And that tells you that the purpose of the condenser is to adjust how much light is actually hitting your sample. Okay, so it adjusts and it concentrates light. You then also have your focusing knobs. And so if we're looking again at the picture, we have the stage, we have the condenser right under it, this circle there, and then we have the focusing knobs over here, being the coarse one, which is the really big one, and the little fine adjustment one. Now the purpose of the coarse adjustment knob, when you move this big knob here, it moves the stage up and down. Okay, so the big knob is for big movement. It will move the body tube or the stage, depending on what kind of microscope it is, up and down, allowing you to bring the lenses closer or further away from your specimen. Now keep in mind, because it's a big knob moving big distances, you want to move the coarse adjustment knob very slowly. Otherwise, you risk cracking the slide. And that's something I'm not kidding about. I have heard the sound of that dreaded crack as a student ends up moving the coarse adjustment knob too quickly. Okay? The fine adjustment knob, on the other hand, which is that little knob within the coarse adjustment, fine adjustment will not move the stage noticeably. You won't get to see big movements. Instead, the fine adjustment knob just fine-tunes the image after you've already started to be able to see it through your lenses. Okay, in addition to those other parts, your microscope also has a body tube, which on some of them will be a very long tube. On others, it's kind of this almost curved shape that connects the ocular and the nose piece. Now, with that body tube, you notice the top portion has the oculars or the eyepieces, which are where you put your eyes. Now, please keep in mind during pandemic times that the eyes are a portal of entry for pathogens, meaning that pathogens can actually get into your body through your eyes. So please be careful of where you're putting your eyes lately. I know that sounds weird, but basically during these pandemic times, do not put your eyes up against the microscope lens without having properly disinfected it first. Now, when we talk about the ocular lenses, please write that it is a 10x magnification. So when you're looking through these oculars, that gives you 10x magnification. Then you have the nose piece a little further down, and that's that rotating circle which holds the objective lenses. And each of those objective lenses will give you some further magnification. Now, depending on your microscope, sometimes the numbers and parentheses will vary a little bit. A lot of the ones we use, the scanning objective is a 4x magnification, then the low power is a 10x, and the high power or high dry is a 40x. The reason why it's called high dry is that it gives you a high amount of magnification, but you do not use any oil in order to see the image for that one. The highest magnification of the microscope, however, the 100x, that one you do use oil. Okay, so that's the difference between 40 and 100. The high dry is 40x without using any oil. The only time you use this immersion oil, where you put a drop or two on your slide and then put the lens into that oil droplet, the only time you do that is with the oil immersion lens. And please make sure to never get oil on any of the other 3 lenses. Now, when you're using a microscope, you have to keep in mind that the objective lens is not the only magnification. Remember, what did we say before? Your eyepiece gives you 10x magnification. So whenever you're asked for the total magnification that you're seeing a specimen with, you have to take into account both the objective magnification, which I put in parentheses here, and the ocular magnification. So over ocular, you can write 10x. That's 10x magnification. Now, that means that your tricky professor can ask you questions related to magnification in a couple of different ways. So make sure you pay very close, careful attention when you're answering a question about magnification, because I can either ask, what is the magnification of a particular lens? So what's the magnification of high power lens? What's the magnification of an ocular? Or I can say, what is the total magnification observed using a particular lens? So I want you to circle total. Circle star, highlight that word. If the question says total, that means you have to multiply the objective times the ocular. So we're going to practice with that on the next slide. Okay, so as I mentioned, we can either ask the question as, what is the magnification of a high power lens? In that case, I'm simply asking the magnification of that particular lens itself, which in the case of high power would be 40x. Okay. If, however, I said, what is the total magnification? The moment you see total, that means all of the magnification your eyes are seeing when you look into that microscope, which means you first get the 10x of the ocular or the eyepiece, and then you get the 40x of the objective. Okay, so in this case, the high power objective. Okay. So now you have to then give the final answer of 400x total magnification. Okay, so magnification of a single lens versus total. Make sure you always include that eyepiece or ocular magnification. Again, if you have any questions or need any help, just contact me and remind. You'll see more of these kind of questions pop up on quizzes, on post labs and whatnot. If you want a little extra practice, just ask me and remind. The last few microscopy terms that I want you to know are parfocal versus parcentric, and then field of view and depth of field. So what parfocal means is the microscopes that you use in microbiology are parfocal, where once you focus an image at one magnification, when you then switch over to another magnification, your image should stay within focus, only requiring a little bit of fine-tuning, meaning once you focus at one magnification and switch to another, you do not need to ever touch the coarse adjustment knob again. You just use fine adjustment to kind of clarify the image a little, but it should stay in focus at all objectives. Parcentric is a similar concept, except it's regarding the center of the image. So what that means is, if this spot here, let's say, is the center of your image at low magnification, then once you go to the highest magnification, even though the image will look different to you, this spot is still the center. Okay, so parcentric means that the center of the image at one magnification remains the center at all other magnifications. Then you have field of view and depth of field. Your field of view is how much of the image you're seeing. So for instance, in this image here, your field of view would basically be the width that you are seeing. Your field of view includes all 3 colored threads. Your depth of field, on the other hand, is how many layers you can see to that image. So at low magnification, you notice you can see all 3 colored threads that are stacked on top of each other. You can see them clearly. Once you go to a higher magnification, what do you notice about that depth of field and field of view? Well, now you can only see the yellow thread clearly. The red and the blue have become blurry. Okay, so your depth of field has decreased. You can only see 1 layer. You can't see all 3 layers clearly anymore. You also notice that your field of view decreased. You can't see all of these long threads. Instead, now at a high magnification, you only see a smaller representation of the field of view. So as magnification increases, your field of view, meaning how much of your image you can see, as well as your depth of field, meaning how many layers you can clearly see, they both decrease as magnification increases. Since that was a whole lot of words, a lot of information, I have rewritten out the definitions that I just gave you, so you can pause the video and jot down these recap notes. Now that's enough of the boring microscopy stuff. You've probably seen it all already. The last thing we want to cover in today's lab is the ubiquitous nature of microbes, which is usually the first experiment that you would get to do in a microbiology lab. Now, before you can understand what ubiquitous nature of microbes means, you have to ask yourself, what is ubiquity? Okay, you hear that a lot in the religious sense. People will say their religious deity is ubiquitous, meaning it is everywhere. So ubiquity or ubiquitous means something is everywhere, which then tells you the ubiquity of microbes means that microbes are everywhere. They're all around you. They are inside of you, on you, in the air you breathe, the food, the water, everything around you has microbes. Now, the cool part about this is we can study or analyze the fact that microbes are everywhere by doing a few very simple experiments in a classroom setting. You can see the results of some of them right here from one of the semesters. All we have to do is use TSA plates. All of these plates are called TSA, trypticase soy agar plates. That means that they are a general media, so they're not selecting for particular microbes, they're not differentiating between them or anything. You'll learn more about that in later lectures and labs. It's a general media, meaning it has everything any kind of microbe, usual microbe, would need to grow and survive. It's got all the food and nutrients that microbes need, whether they're bacteria or fungi. So TSA plates are basically used to grow a whole lot of different types of microbes in robust, good amount quantities. So the way we use these TSA plates to study or analyze the ubiquitous nature of microbes is by doing air sampling of the air around you. We do hand prints, and we used to do mouth prints or mouth swabs, but that won't really be allowed anymore due to the changes that the COVID pandemic made universities see. And then we do fomite testing, which means we swab different items or objects in the classroom. Okay, so to study or analyze the ubiquitous nature of microbes, you can do air sampling, you can do body sampling, which would be the handprint or mouth swabs, or you can do object sampling, which would be fomite testing. That's Fomite, and you're going to see that in the upcoming slides. Now, the first type of quality control sampling that you can do for the ubiquitous nature of microbes is air sampling. And when you're sampling the air around you, it can either be active or passive. And think about those terms. If someone, even you, if you are active active, what does that mean? Got a lot of energy, you use a lot of energy, right? Whereas passive, just chill, let things happen, right? So the difference between active and passive air sampling, active air sampling requires energy. It requires special equipment that will pump air through a piece of equipment, computerized equipment, that will then read off or allow you to find out what kind of microbes are in that air. And you see that in the pictures down here. Okay, so any active air sampling will have a special piece of equipment with an internal pump to pull an air sample through to analyze it. Whereas passive air sampling is what you get to do in the classroom. All we do is we take one of those TSA plates and we simply open it up in the classroom. Let it sit for an hour or 2 hours. Basically, it depends on how long your class is. Sometimes it's as short as 20 minutes sitting. Sometimes it's as long as 3 hours waiting. But basically, it's however long you have to let that plate stay open in a particular environment, picking up whatever microbes it can in that time. Now you got to ask yourself, well, what are the pros and cons of each method? Why might we want to use active? Why might we want to use passive? Okay, in terms of, your different functions. So first, let's talk about active air sampling. That's the one with the big piece of equipment, the fancy piece of equipment, pulling air through. And as soon as you hear special equipment, fancy, well, the pros of that is odds are that pump is going to get every little bit of air. You have a better accuracy with that experiment. So active air sampling has the pros that it's more accurate. You are more likely to get a good amount of microbes because you're forcing the air into that machine. And it's nice and quick. You don't have to leave a plate out somewhere. You have that machine capable of pumping or pulling the air through. So it is quick and accurate. Now the cons of it is, well, what did we say? It needs a fancy piece of equipment. So it means you have the con or the negative that you need special equipment, which requires special training. And when you see fancy equipment, what does that tell you? Expensive. Okay, so it's more expensive. Once you know those pros and cons of active, it means it's a lot easier for you to figure out the pros and cons of passive air sampling. So in our classroom or in a lab where you're just going to leave an open plate out to collect bacteria or fungi, well, the pros of that, you don't need any special equipment or any special training. You open up a plate, leave it there. Okay, so it's easier. And without special equipment, it means it's cheaper too. One of those little plates does not cost much compared to a big piece of equipment. Okay, so passive air sampling is easier and cheaper. The downside is you're relying on gravity in a small area. You can't get every microbe in that room. So it's going to be less accurate. It's also going to be more time consuming because you have to leave it just sitting there hoping that whatever bacteria or fungi are in that room will find their way onto the plate. Okay, so passive air sampling is good in that it is cheap, it is easy, but it's bad in that it's not going to be as accurate and it's going to take a long time. You need to leave that plate for hours before you'll actually see anything. But because it is cheap and easy, that's why the answer to which are we performing? Obviously passive. We want cheap and easy for a classroom setting with students. You also usually get to do handprints and mouth swabs. Now, when you think of body swabbing like handprints or mouth swabs, your skin is basically covered in tons of microbes. And I know you think, ew, gross, that's nasty. But in fact, you're going to see in lecture, that's your normal flora. A lot of the microbes that are covering your skin and that are in your mouth all over your body, they're actually helping you. they're beneficial. But because everyone may have different normal flora and because you're encountering different things throughout your day, you end up having the issue of contact transmission being so common. Now, when we say contact transmission, that basically means you are touching something that ends up making you sick. You are getting sick usually from contact with a sick person. Now, there are three main forms of contact transmission. There's direct, there's indirect, and there's droplet contact transmission. So direct, indirect, and droplet. Direct contact transmission is things like kissing someone, shaking hands, hugging, even sexual contact, or even an animal bite. That's direct contact transmission. Indirect contact transmission is, let's say you are a nurse or you are a parent or a loved one taking care of someone who's sick, and you touch their used tissues to throw out, you touch their thermometer that's been in their mouth, you touch any of the items that a sick person has used that you associate with a sick person. The last one is droplet transmission, droplet contact transmission, and that has become very well known to people throughout the COVID pandemic. Droplet transmission is when you inhale or take in droplets that have microbes in them, either from someone coughing, sneezing, or even when you speak and those little fine particles of droplets come out of your mouth. Now, these forms of contact transmission are different from vehicle and vector transmission that we'll go more in detail in lecture. Vehicle transmission, picture vehicle as car. Basically, a microbe is hitching a ride on something that you would normally not think of as being associated with a sick person. So, for instance, you end up touching a doorknob handle, or a pen, a pencil, a desk, any inanimate object, or foamite, which we'll talk about in the next slides. You touch this inanimate object that you never saw a sick person touch, but it happens to be contaminated with microbes. Okay? And that's the difference between vehicle transmission and indirect contact. In indirect contact, we were talking about inanimate objects that you associate with a sick person. You saw, used tissues, thermometer, okay? Whereas vehicle transmission is inanimate objects that happen to be contaminated that you wouldn't know. So things like a doorknob, pen, pencil, cell phones. The last one is vector transmission, and vector transmission means that you got a microbe from an arthropod, meaning insects or arachnids, or things like fleas, ticks, any of those. That would be vector transmission. The big example being mosquitoes, which infect a lot of people across the world with different infections. And we'll talk more about that in the lecture. Now, when you do these swabs, you are focusing on contact transmission. The idea that your hand or your mouth is coated with all different types of microbes. Now, I want to ask you, if you were going to do this experiment in lab, which do you think would have more growth, a handprint or a mouth swab, and why? I'm going to have you write about that in your post-lab, kind of thinking about, well, a handprint, all you do is you take your hand and you put it on the TSA plate to transfer whatever is currently on your hand. A mouth swab, you're swabbing the inside of your mouth, your cheek, and then transferring it to a TSA plate. So which area do you expect to have more variety, more growth of different microbes? Your hand or your mouth? And think about why that is. Okay, so again, that was a lot on the previous slide, so I've given you some recap notes. You can pause the video to jot down some notes. You'll see this again in lecture. And in these recap notes, I even specified the two different types of vector transmission that you can encounter. So whether it's mechanical or biological, each one is still occurring through an arthropod or an insect. Okay. So the last test that we do to kind of demonstrate the ubiquity of microbes, the fact that they are everywhere, is to test fomites. And fomite is a fancy way of saying an inanimate object, okay, meaning not living, an inanimate object that may be contaminated with microbes. And examples that you use in everyday life are things like your pens, your pencils, your cell phone and your keyboards especially. Even things like a shoe or pieces of clothing that you might touch can be contaminated, and doorknob handles too are another big one. Now, what kind of transmission did we just say this is? This would be vehicle transmission. Okay, so these are inanimate objects that have been contaminated by microbes hitching a ride on them, and they can make you sick. And the way, again, that it differs from others is the fact that you would not associate this with a sick person or an infected person. Okay, so don't get vehicle transmission mixed up with indirect contact. Again, if that's not clear to you, please send me a message and remind, and I'll go over it a little more. Now, the last part of this lesson, I want to give you a few sample results, and I'll have you analyze them in your post-lab. Because honestly, when it comes to laboratory work, anyone can follow a protocol. I tell you to put this much liquid into this particular tube. Yeah, great. But what really is important in being a scientist is being able to analyze results. What do your observations actually tell you? What do they mean? And that's a skill that you're not just born with. You have to practice this. So throughout these online lessons, I'll give you sample results that I'll then have you analyze in your post-lab. The first one is showing you a result in which you're looking through your microscope and you see this image. And in your post-lab, you'll be asked about what does that result tell you. The second result is focusing on a variety of different handprint results from one of the semesters. Okay, so these are actual handprint results, and you'll notice that not all of them look the same. There are various different types of growth. When you look at handprints, you'll notice the small, smooth, shiny, circular type of growth is usually bacterial, and some yeast will also grow that way. Whereas the fluffy, flower-like, spreading out growth, things like this blob here, anything fluffy and flower-like is usually mold fungal growth. Okay, so smooth, shiny, nice, defined little circles are usually bacteria, or in some cases yeast fungi, whereas fluffy and flower-like is mold. And we'll go over that more in lecture. And so in the post-lab, I'll have you analyze these results and why you think this may have happened that not all handprints look the same. What can affect the growth and variety that you end up seeing on a handprint? Keep in mind the way that these handprints are performed is that students are told to open up the TSA plate, meaning remove the cover, take your hand, usually your dominant hand, and simply press it against the agar for a few seconds. Now, usually that agar is a little cold, there's some moisture, a lot of students get grossed out. So keep that in mind when you're analyzing the results, because how might that affect how they did their particular handprint, okay? As well as other factors that you can think of. Now, when you do post-labs and when you're analyzing any kind of results, please don't try to Google answers. Not only is that cheating and can result in your failure in the entire course. I don't just fail you for the one assignment, I fail you for the entire course if you cheat. But it also takes away from you because I'm not looking for one particular right answer. In an analysis like this, I'm basically looking to see what your logical thought process is, okay? So you're not going to get penalized when you're analyzing something like this where I'm asking you for what you think are some factors. Okay, I didn't give you any answers in the narration, so you're not graded on that. I just want to see your logic in analyzing this particular result. Result 3, these are air plate results, real air plate results. Okay, so students opened up, removed the cover, and left that agar sitting in different locations. Okay, one of them picked a bathroom, one of them picked the stairwell, one picked inside the classroom, various different places, but all I'm telling you is that one of them was placed in a room in our building, this was at a different campus, where the owner of that room kept rescued kittens and cats, meaning that there were animals in there were litter boxes, there was cat food and everything in there. So looking at these results, all of these different plates, I want you to try and figure out which one do you think was where the animals were. And the way you interpret plates is you look for growth. The clear beige, that's the agar itself, and then you look to see, are there spots, speckles, fluffy growth, which we just mentioned, a change in any color, okay, to try and see what kind of growth you're seeing. Okay, again, this is just to see where your logic is, kind of have you doing a little thinking before we get to the real experiments in later weeks. Results number 4 is focusing on fomite swabs, and basically what the difference in terms of the two different halves of each plate is that when the fomite, meaning the inanimate object such as a sink handle or a pen or cell phone, when the object was swabbed, it was either swabbed with a wet swab, meaning the swab was dipped in water first and then swabbed, or after that wet swab was collected, they then took a dry swab and swabbed the same fomite. Okay, and on some of the plates we noticed the wet side had more growth, and others we noticed the dry. So I want you to think about some reasons why that might be. So keep in mind how I just told you the experiment was performed. So wet swab first, then dry swab. And also think about what you know in everyday life of things like moisture versus dryness and microbial growth. Okay, again, this is just an exercise to kind of get you thinking, start analyzing. In later weeks, there'll be more of a correct answer versus incorrect answer. This week in these kind of result analyses, it's more just to see your logic. Okay? Again, if you have any questions, any concerns, you need help with even the post-lab questions, because post-labs are not quizzes. Post-labs you are allowed to look at are lecture slides and my narrations, okay? You're not supposed to be using internet outside sources, just the lecture slides, the narrations for this. Whereas quizzes are when you won't be allowed to look at any of our notes. Okay, and quizzes will be timed, post labs are not. You have the whole week to do the post lab. You're allowed to ask me questions as you're working on your post lab if you're unsure of anything. If there's any slide you would like me to go over in more detail, please just send me a remind message. I do not mind. You're never bothering me. Just simply tell me which slide you're referring to, and I can help you out. I can also give you practice problems to help you